Hi everyone, welcome back. So today we're going to be learning about DNA replication. So before we get started, I actually have a YouTube video that I want to show you. It's very short, only about 4 minutes long. So a couple of things to keep in mind when you're watching this YouTube video. As it goes through the process of DNA replication, first off, we need to remember that DNA replication happens during the S phase. So DNA is still in the nucleus, it cannot leave the nucleus, and this is when replication is going to occur. So this process is going to be done in order for cells to eventually make another copy, which happens during the cell cycle, which we already learned about. So during DNA replication, you have several enzymes that are working in order to make this copy of DNA. So if you remember back from biomolecules, enzymes end with the suffix ase. So helicase, ligase, nuclease. You're going to hear about some of those things in this video. Not only that, there's extra enzymes here that we're not actually going to talk about in our notes. There's so many enzymes that work together for DNA replication, and scientists are actually discovering more all the time. So on our notes, we're just going to focus on the main ones, but in the video, they are going to discuss a couple more than what we'll be talking about. So let's go ahead and get started. My computer froze. There we go.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, so let's go ahead and hop on over to our notes. So hopefully that kind of, illuminates a little bit how DNA is going to be replicated. So let's go ahead and write down some of that information. So how does DNA make a copy of itself? So we know that this is a process that occurs in the nucleus. Okay, so DNA is actually pretty fragile. It cannot leave the nucleus. And so later on, next week or during the week, depending on when you watch this video, we're going to talk about a process called protein synthesis, and that gives us the mechanisms to transmit that message from DNA to where it needs to go in the ribosome. But we'll talk about that one later. So, and I actually just realized I switched this. Okay, so the location is in the nucleus, and up here it occurs during the S phase. Okay, so S phase of cell cycle. I'm recording this in the morning. I think I need another cup of coffee. So when we're talking about semi-conservative, that means that our new strand has half of a new strand and then half of an old strand. Okay, so newly replicated DNA has half, old strand plus half new strand. Okay, so that's what it means by semi-conservative. And you can see in this picture down here, so my blue DNA, my blue strand of DNA, because it splits apart, the blue strand is going to be my old strand. This red strand right here with the red backbone, that's going to be my new strand. Okay, so each of these is going to be two pieces of DNA, and half of it's old, half of it's new. Okay. We also learned about several enzymes that are involved with DNA replication. So just like I told y'all before we watch the video, we're not going to discuss all of them. We're just going to discuss 4 of the main ones. So first off, okay, so we have DNA helicase. Okay, and I'm actually going to underline this. Okay, so DNA helicase is the enzyme that's responsible for untwisting our DNA. So we say this one unwinds parent DNA strand. Okay, so remember, we always start with a parent, just like how we started with a parent cell during the cell cycle, and then we end up with daughter cells. So we start with a parent strand and we end up with daughter strands. And you can see that right here in the picture. So we have 2 daughter molecules down at the bottom. So we have DNA helicase. Okay, so DNA helicase is going to unwind that parent DNA strand. We also have DNA, okay, so DNA polymerase, sorry. Okay, so this is the enzyme that adds on free nucleotides. Okay, so remember, our nucleotides are going to be that phosphate group with the sugar with the ATGC. Okay, so phosphate, sugar, and a base. Okay, so our DNA polymerase is the enzyme that adds those on. All right, we also have primase. So DNA polymerase needs to know where to bind. So primase adds a small RNA primer. So DNA polymerase knows where to begin. Okay, so that primase is just going to add on that small little RNA primer. So that way, it just marks, okay, this is the starting point. This is where we're going to start replicating. All right, so we have primase, and then lastly, we have ligase. Okay, so ligase seals our DNA backbone. Okay, Okay, seals or glues the DNA backbone so it is continuous. Okay. So those are the 4 enzymes that we really need to know about. Okay, so DNA helicase, which is going to do the unwinding. Polymerase adds on our bases. Primase, you have that small RNA primer that is the starting point, and then ligase seals up everything. So just like how I had like a little hand gesture for mitosis, I have one for DNA replication, so that way you know the steps. And it's a lot more simple than the mitosis one. So first off, okay, so if you have your arms crossed, twisted, okay, so this is my strand of DNA. I'm going to first unwind. Okay, so that's my first step. Create a new strand, and then twist it back up. Okay, so those are my steps for DNA replication. Okay, so unwind, new strand, twist it back up. All right, it's pretty simple. So, all right, let's go ahead and do some labeling. So what I have here, this is a replication fork. Okay, so just like how you have a fork in the road, okay, this is a replication fork. So you can see I went ahead, I already put a box here for single-stranded binding protein. Something you don't need to know. You may see it on a diagram, but that's more of an AP thing. Same thing with topoisomerase. Okay, so topoisomerase, again, it's another enzyme you don't need to know. All right. So here are my DNA strands, and I can see now it's going the opposite direction. So I'm going to go ahead, I'm going to start, adding in my little drag and drop so that way we can figure out what goes where. So first off, I can see that this right here, okay, so I have this enzyme that's unwinding, it's splitting apart my DNA. So if we recall from our last slide, that's our helicase. Okay. So I also have this guy right here. Okay, so this big orange box, that's another enzyme. So I can see what it's doing. It's moving in that direction. And I can see, as it goes past, it's added on new nucleotides. So I know this enzyme is going to be my polymerase. All right, so next, okay, I have another strand. Let's see here. I have right here. Okay, so this one up here at the top. Okay, so it kind of looks like a donut that's on one side of my DNA, on my DNA strand. And so with this one, it's going to be adding on my RNA primers. I can see there's an RNA primer on the opposite end. Okay, so that one is going to be my primase. Do a little searching. Okay, all right. So lastly, let's see here for this top enzyme. Okay, so I have this enzyme that's right here. Here's my another DNA polymerase, because DNA polymerase has to work on both sides. And I can see that this little enzyme looks kind of like a flower. It's traveling along the backbone of the new strand. And so I know for that one, okay, that's my DNA ligase. Okay, so my DNA ligase essentially seals up my Okazaki fragments. So Okazaki fragments are the short sections of DNA that are going to be laid down just because, we have one strand that has to go backwards. So this bottom strand right here can be formed continuously. So once your DNA polymerase attaches on, it's going in the direction that the DNA is unwinding. And so it's just going to continually add on bases. And so the other side, it has to do it discontinuously. So essentially, because the DNA is going backwards, DNA polymerase has to attach and then add on like just little short strands, just because, it is going backwards. And so those little short strands are called Okazaki fragments. Okay. So if you have an Okazaki fragment, we call that one the lagging strand. Okay, so lagging strand, it's going a little bit slow. And so, those Okazaki fragments are being formed, and eventually we're going to have ligase seal those fragments together. So one that can go continuously is going to be our leading strand, because once that polymerase attaches on, it can just keep on going. So lastly, I need to fill in these 2 little boxes. So I told y'all in the last video that DNA runs anti-parallel. So what that means is that if I start with a 3' end, okay, and I trace it, okay, then that means that this side is going to be 5'. Opposite, if I start at 5' over here and I trace this one back, Okay, I can see it ends at 3 prime. And so our newly synthesized DNA strand is going to do the same thing. So let me go back over here so I can know which boxes to fill out. Okay, so I know that this one right here is 5 prime, and it's not letting me type. Is there a number that I can drag? Oh, there are. My picture was covering it. Okay, so there's my 5 prime. And then because I know it's running anti-parallel, that means that the opposite side has to be the opposite number, which is 3 prime. Okay. All right, so let's go ahead and put these steps in order. That way we know the steps that DNA replication occurs. Okay, so first off, we know that we have our parent strand of DNA and that it's going to have to unwind. And so first, let's see here. Okay, so first can actually be one of two things. So we can actually either form a replication fork, but I'm going to put DNA helicase for my first step. It's going to unwind that DNA because once it unwinds, that's when you get that fork. Okay, so DNA helicase is going to unwind my DNA, and then a replication fork is formed. So next, let's see here. Okay, it says DNA polymerase attaches to the primer. Okay, so do I have anything here about primase? Let's see here. I don't. Okay, so normally, I could have another step in the middle saying that primase is going to attach a primer, and then DNA polymerase will attach to that primer. So once DNA polymerase attaches to that primer, my DNA polymerase is going to add nucleotides. And then remember, in the lagging strand, we're going to have Okazaki fragments. And then the Okazaki fragments have to be bound together with ligase. All right, so let's go ahead. We're going to test our knowledge. Okay, so right here, go ahead and pause the video, okay, and go ahead and try to fill this out. And then I will be back so that way we can check our answers. All right, hopefully you paused. Okay, so let's go ahead. Let's see if you got it correct. Okay, so the enzyme that unwinds DNA. Okay, so we know that's DNA helicase. Okay, so sections of copied DNA that's created on the lagging strand. Okay, those are my Okazaki fragments. Okay, the strand that is copied in a continuous way is my leading strand. Okay, binds Okazaki fragments. DNA ligase. Builds a new DNA strand by adding complementary bases. Okay, that's my DNA polymerase. Okay, stabilize DNA molecule during replication. Okay, so that one, I'm going to say that's my replication fork. Okay, so, oops, not form, fork. Okay, so just because when it's unwound, okay, it's going to be stabilized. Okay, the strand that is copied discontinuously, that's my lagging strand. And initiates the synthesis of DNA by creating a short RNA segment. Okay, that's going to be my primase. Okay. How'd you do? Hopefully you got all of these. So if you have any more questions, definitely look at the end of this presentation so that way you can have a list of videos in order to watch just in case you need a little bit more assistance. Definitely ask your teacher if you need help, ask me, ask Coach Richie if you're one of his students, and just hopefully you understand this. So I hope you all have a great day and I'll see you next time. Bye.